This is the last chapter that we're going to go over before your exam number 2, so let's get started with Chapter 8, Special Senses. And if we think about the special senses in our body, we know that there's 5 of them. We have touch, taste, smell, sight, and hearing. And there's actually one of these which is a mixture of the general senses, thinking about temperature, pressure, and pain, and that's going to be touch. Now, if we look at a subset, we can also think about the special senses, and these include smell, taste, vision, balance, and hearing. And all of these special senses we're going to cover throughout this entire chapter. We're first going to start off with one of the most important special senses, and that's going to be the eye and vision. And it's actually 70% of all your sensory receptors are found in your eye. And because these are so important, taking up a lot of the sensory input, each eye has over 1 million nerve fibers. So think about all that sensory input towards your occipital lobe. Now, what function does this have? Well, all of that information is from the eye is going to be sent to the brain. Now let's think about protection for the eye. And so if we look at the eye itself, it's housed, if you remember your skeletal system, we have that orbit found in the eye. And so the eye is enclosed in a bony orbit. And we also have a cushion of fat surrounding most of the eye. And what's also interesting is if you look at the eye, so here's an example of someone's eye, the eye appears like we only see 1/16 of an inch, but in actuality, the eye in itself is 1 inch in diameter, but only that first 1/16 anterior region can be seen, so pretty interesting. Now looking at accessory structures of the eye, these are components that help protect or shield the eye from damage, and we have the eyelids and eyelashes. We have what's called the conjunctiva. Oops. We have the lacrimal apparatus. And we have extrinsic eye muscles. In this first illustration, what do we see? We see the eyelid. And this is that first layer helping shield and protect the eye. And then we have the eyelashes, also helping shield and protect the eye from the sunlight. The conjunctiva, this is that inner layer, so right underneath your eyelid is the conjunctiva, and it secretes a mucus to help lubricate the eye. Conjunctivitis, this is an inflammation of the conjunctiva. And for anybody who's ever gotten pink eye or know of someone who's gotten pink eye, what is that? Well, that's when you have a bacterial infection within your conjunctiva. And also a common way of getting this is not washing your hands and scratching your hands on your eye. And you remember we have lots of moist membranes. And here, this conjunctiva, because it secretes mucus, this is a nice mucus membrane. And because of that, it's this easy barrier anytime you have a pathogen, bacteria, virus, or anything. Scratching your eyes, having contaminated hands, that can easily allow any of these pathogens to enter into your eye. Now, one way common in getting pink eye also is if you use a restroom and you don't wash your hands and then you touch those doorknobs, door handles, and you scratch your eye, you can easily get, if somebody used the restroom and after they have feces on their hand and they touch the surface and you touch that same surface, getting that in your eye too. So very common. Also for women, it's also important is because you don't want to be sharing your eye makeup. So you can catch bacteria. If someone has bacteria within these eye makeup, you can spread that. So try not to share your products. You can always transmit that bacteria. Now we have this other structure here called the lacrimal apparatus, and this deals with your lacrimal gland and the duct system. And the lacrimal gland, this is going to produce all of your tears. And so how does this work? Okay, so your lacrimal gland is going to produce all of your tears. It's going to enter here into this lateral region called the lacrimal canaliculus. From here, your tears are going to drain into your lacrimal sac, and then that can then be expelled into your nasolacrimal duct into your nose. Okay, so we can continuously regenerate this lacrimal gland. Now, other things too. So if you notice, your nasal cavity is connected towards your eye. And so I'll post a video for you on YouTube. There's a famous painter who actually sucks up paint into his nasal cavity and then squirts out that paint into this, so it comes out of his lacrimal sac into this lacrimal duct, and it comes out of his eye. So that's how he paints. It's pretty nasty. And you probably know some people who also suck up milk through the sac and can squirt their milk out of their eye. So pretty gnarly stuff. Okay, watery eyes, that can happen is if there's a clog or blockage here within this lacrimal canaliculus, and that can give that overproduction of water, tears coming out, or also if your lacrimal gland is producing too much tears. Sniffles, how does that happen? Well, if someone's crying, why is it that not only do their tears start coming out, but you start having a stuffy nose or runny nose? And once again, because we have drainage. Same thing also when you yawn. So I know I'm addressing a lot of things because I always get these questions every year. And so when you yawn, what happens? Well, as you yawn, you can start putting pressure against this lacrimal sac, and that in itself causes the tears to come out, or also causes that lacrimal sac to empty into your nose. Okay. Other accessory muscles or accessory structures we have, these are the extrinsic eye muscles, and we have several of them. Here we have the inferior oblique, once again being this angular, so going at an angle. So inferior oblique, this is going to elevate your eye and turn it laterally. Okay, as this contracts, it's going to pull towards that direction. Okay, other ones we have inferior rectus muscle, and rectus meaning straight. So this is going to elevate your eye and turn it laterally. Here we have lateral rectus muscle. This is also going to move the eye laterally. As this contracts, it's going to move it in this direction. Superior rectus muscle, also the straight muscle. As this contracts, it's going to elevate the eye so it can go in that direction and also turn it medially. And then superior oblique muscle, so this one is going to turn it laterally and also elevate the eyes. Medial rectus, we couldn't see that too well in the other picture, but here's that medial rectus. Rectus being straight, and notice as it contracts, it's going to turn the eye medially. And here's that superior oblique. Notice contraction. As this contracts, it's also going to point the eye towards that direction. Now if we look at the anatomy of the eye, here's a cross-section of it, and the eye itself is composed of lots of structures and lots of layers. And so rather than going through each one by one, we're going to go through the anatomy and the function of each of these one layer at a time. And so we form 3 different layers in our eyes. We have the first is the fibrous layer. And that's this outermost layer that we see here, white, and also the most anterior, most clear region. We have the vascular layer. Vascular layer, we see that here in red, all that nutrient blood supply. Then we have 3, the sensory layer. And that's going to be this innermost layer that you see here in yellow. Gives rise to our vision. Let's start off with the very outermost layer being the fibrous layer. And the fibrous layer is composed of 2 major regions. We have the sclera, that you can see here in white, so it's that white of our eyes. And the function of this is for protection. of all these underlying cells. Anteriorly, we have the cornea. It's that clear structure. And the function of this is to allow the passage of light through and into the eye. Repair, because this is a surface that we constantly comes into contact with external environment, whether you're rubbing your eye, whether you're using contact lenses, this is always going to be undergoing abrasion. And because of that, this cornea constantly gets repaired. And actually, what's interesting is that the cornea is the only tissue that can be transplanted into any human being without fear of rejection. So it's only tissue that won't be rejected. Next layer we have is this darker layer over here, and this is the choroid layer. Choroid layer, this is that nutritive, so it's blood rich and nutrient rich layer. So this is the one that gives all of that oxygen and nutrients to our underlying cells and tissues, and as well as to our sclera. More anterior, here we have the ciliary body. Ciliary body can contract, and by doing that, it can regulate and bend our lens. Ciliary body, this is composed of smooth muscle. Now, anterior to this, we have the iris. And the iris, this is actually going to regulate the amount of light in and out of the eye. Okay, and so cornea is just that passageway for light, but the amount of light coming in is all by this iris. So this iris is going to control this region over here. And pigmentation, and so your eye color, all of that is determined by this iris. So it's all on your iris. So this gives rise to unique eye color. Now here, as we mentioned, this iris can regulate the amount of light entering and exiting your eye. So it can dilate, in which case this region between each of the iris is going to be, as well, the cross-section of this iris. If we look at this cross-section, this can be wider, have a larger or wider pupil. And if it contracts or constricts, then we can have a smaller pupil. And so this pupil, what this is just the hole or opening that's controlled by the iris, where light enters the eye. Now, I just want to show you this really quick. So all this light is coming in, and when we get to this in the next slide, we're going to go over the innermost layer, being the sensory layer. But if you notice, all of this light is going to start shining towards this region right here. And I just want to show you, this is called the fovea centralis. And this is this region that has the highest visual acuity. And this is why we're always trying to regulate the amount of light entering and exiting our eye, because we want to focus all of that light towards this region, because this gives us the most vision. Okay, or acuity for our vision. Okay, next area we're going to look at is this iris, a closer look at this. And the iris is actually composed of two different types of smooth muscle fibers. We have what are called circular and radial muscle fibers. Okay, so here we have circular, and once these are contracted, we're going to have a smaller pupil. And when do you think you're going to have a smaller pupil? when in response to lots of light. Now you're going to contract your radial muscle fibers, in which case you're going to increase the size of the pupil. And this is in response to dim or darkness. So dim light or darkness. Now also, what do you notice? If you ever drive a car at nighttime and then you pass by a cat, you'll notice that the moment your high beams or your headlights hit that cat's eyes, the cat's eyes are very dilated. And why is that? Well, in any event that you don't have a lot of light, you're going to start dilating your eyes to allow as much light as possible into your eye, and that's why we dilate our eyes. Now this is under autonomic or somatic nervous system control. Okay, this is under autonomic. Good. Somatic deals with our skeletal muscles. Autonomic deals with smooth muscles. Okay. And also, if we consider if you're in a fight and flight versus rest and digest state, the moment that you are in a fight and flight situation, if you want to flee a situation, most likely your eyes are going to become dilated. And why is that? Well, you want to be more observant, take in more light of your entire surroundings. Now the next layer we have is the inner sensory layer, and this is composed of our retina. Our retina in itself has two layers, and it has an outer pigmented layer. And we have an inner neural layer. And I want you to keep a note of this fovea centralis because we're going to get into that. Now let's take a look at this inner layer. This is the inner neural layer, and it's composed of two different types of cells. We have rods and we have cones. So cones you'll see here in yellow, and rods you'll see here in purple. And cones, I remember them because these are for color, and rods, these are for your gray tones. And so everything from white all the way to black. Now, what is the function of these rods? These rods, they allow dim light vision and peripheral vision. Okay. And then what's also interesting, so as you approach or go closer towards the eye, you're going to find a lot of rods within this area over here. So it's more anterior, surrounding this lens of the eye. Versus the cones. Cones are going to be centralized right over here, and we call the fovea centralis. This is where we're going to have the most, so it's the area of the retina where we only find cones. And because of that, this is the area that's considered and gives rise to our visual acuity and our sharpest vision. Okay, and an ability to view things very critically. Now, a couple of things. So let's go back to rods. Okay, rods are going to start taking over, especially in dim light, low vision. And so at night, if you notice your light is on, and before you go to bed, you're going to turn the lights off, and the room seems completely pitch black, really dark. But after a while, you start accommodating to this darkness. Your eyes are going to start dilating, your pupil will start dilating, and then you'll be able to see within the dark to a certain extent. And why is that? It's because of these rods. They're going to start discriminating between the light and the dark shades, and that's how you can start seeing later at night. Now, if somebody does not have, or if they're deficient in vitamin A, this can give rise to what's called night blindness. In which case they can't see within the dark or dark regions. No. How does this work? Okay, so once we receive, so we have light entering here first through the cornea, then we're going to enter here through what's called the lens, and we're going to use that lens to focus all of that light at that fovea centralis. And of course, we're going to start stimulating our rods and our cones. And then from here, our rods and our cones, if and when they're stimulated, they're going to send that signal down to what are called bipolar cells. Bipolar cells are then going to send their signal down to ganglion cells. And these are both types of neurons. Just different terms. Now here we have the dendrites, here's the cell body, and here is the axon. And so once we get stimulated, we're going to send this message down the entire axon of those ganglion cells. So here's a cross-section of that, looking at this illustration down here. We're going to send that axon down here, all the way out of our eye, until we're going to reach one lobe of our brain. Which lobe are we going to try to reach? Our occipital lobe. Because remember, occipital lobe is where we have all the interpretation of our vision. And so here, all you're going to see at this most posterior part, where all these axons leave, we're going to see what that's called. That's called the optic disc. Okay, and because this optic disc is filled with axons from all those ganglion cells, this is an area where we don't have any of those photoreceptor cells. Because we don't have any photoreceptor cells here, we can have no interpretation and no stimulation of sensory neurons. Therefore, we also call it the blind spot. So the optic disc is also called the blind spot. And so therefore, once again, we cannot process any images on this optic disc. And why is that one more time? Because there's no photoreceptor cells in this area. Now, what does this form? Remember, if we had that connective tissue surrounding all of these axons, all those axons collectively form nerves, and the nerve that's going to leave our eye is going to be the optic nerve. Okay. So looking at our cones, there's actually 3 different types that take in 3 different wavelengths of color. And so we have the blue cones, so we take in wavelengths of light from about 380 to 475. Green is going to take in colors from wavelengths of around 450 to 625. And then reds, this is going to be including a large range. So wavelengths from about 450 all the way to 660 or 675. Now, what about intermediate colors? And so let's take a step back. Anytime any wavelength of light is absorbed within this region, it's going to come out and appear to us as blue. Here, appear to us as green. Here, appear to us as red. Now, what about intermediate colors? So anytime you see something as purple, how does that look like? So intermediate colors, that's when impulses are received at the same time from more than one type of cone. And that's what those intermediate colors are. So let's write that down. These are impulses that are received at the same time. From more than one type. Of cone. And as an example, we had purple, which is when blue and red cones are both stimulated. Now, what about color blindness? Okay, so there's 2 different types we're going to look at, and that's partial color blindness, and this is if you lack one cone type. And there's total color blindness, in which case you lack all 3 cone types. And what is common and what's seen often is the lack of red cones, and this will give rise to red-green color blindness. And so someone who does have this, both red and green are seen as red. Now, color blindness is also considered a sex-linked condition, and it's carried on the X chromosome, and that being the case, it occurs most exclusively in men. And let's just take a quick second. This is the Punnett square, which I'm going to go over. This can determine whether or not you can pass on certain traits or not. You do not need to know this for your exam, but it's just something it's useful to know if you ever take a genetics class. And so anytime if you look at, if you want to see the probability of something, if it's a sex-linked, if it's going to be passed on to the next generation. Here, XY, this represents a male. XX represents female. Now let's say that here with female, if one of your mothers is a carrier for color blindness, we'll just denote that as a C. So C means a carrier for color blindness. And if you have a father that's not colorblind, what is the chances of your child being colorblind? So let's look at first the child. So here we have a Y and an X. Second one, we have a Y and then an X with a C. Let's look at this down here. So female XX and then XC. Now here, if you look at the male population only, the males will have a 50% chance of being colorblind. Why is that? Well, the Y won't have an effect on it because this chromosome for being colorblind is only carried on that X. And that being the case, if that male has that carrier on the carrier trait on its X chromosome, he will express color blindness. This is why it's going to be almost exclusively in males. Let's look at this in females. So here is a female. XX denotes female. Here, this female is not going to have color blindness at all. And here, because the female has 2 X's, here where this female is only going to be a carrier and it's not going to express the trait for being colorblind. Only and if only this female has 2 C's will this female be colorblind. But since it only has 1, it's also considered a carrier, and therefore it won't express the trait of being colorblind. Okay. Now looking at the chambers of the eye, we can notice here we have the anterior chamber, so this is anterior to your iris and also your lens. Posterior chamber is back here. And if you look at the anterior chamber, it's composed of this liquid found in here, and this is called aqueous humor. Now, what is in this? So it's similar to blood plasma. Also, what's similar to blood plasma we saw in the last chapter is that cerebrospinal fluid. And so just like that, it contains all those nutrients, ions, oxygen to help give nutrients to the cells in our cornea and also our iris. So it helps maintain, in addition to that, it's going to help maintain what's called intraocular pressure. And intra meaning within or inside, so it's this pressure within the eye. So it prevents this from collapsing. And it also provides nutrients for the lens and the cornea. Okay. Just like that cerebrospinal fluid, we're going to continuously replenish all this aqueous humor. And so after this is produced and we can cleanse this out, all of the aqueous humor is going to drain out through this little dot that we kind of see here, but this is actually a canal. And this is called the canal of Schlemm. And if any time this canal gets blocked up or clogged or anything like that, what's going to happen to the pressure in here? The pressure is going to start increasing, in which case that's going to give rise to glaucoma. And so glaucoma, what results from that is a high intraocular pressure resulting here within this aqueous region. And that in turn is going to put pressure on your lens, on this posterior cavity. That in turn is also going to put pressure on all of your inner sensory layer that we see here in yellow, and that can cause damage to your sensory layers, so your sensory cells, and also impair your vision. posterior chamber we have is so here's posterior chamber. We also have a humor. This is called a vitreous humor. We can kind of see that here in that opaque white. It's a gel-like substance posterior to the lens, and the function of this is to help maintain intraocular pressure. Okay, so this aqueous humor and the anterior chamber helps maintain the intraocular pressure over here. And then we have the posterior chamber filled with vitreous humor to help maintain the intraocular pressure in the posterior region. Now let's go over vision. So we went over the 3 different layers, and we're going to see how those 3 layers work in creating vision, and then also the use of the lens in aiding in this process. And so let's first look at this pathway of light through the eye. First, all that light's going to enter through our cornea, through that aqueous humor, and then to our lens, back to our eye. So as we can see already, there's lots of factors that's going to cause the speed of this light to change by the time it enters from our eye to the time it actually reaches that fovea centralis. Okay, so a couple of factors we have air, we have the cornea, we have the aqueous humor, the lens, the vitreous humor. Okay. Now, we need to focus all that light on that fovea centralis for that optimal vision. And so, how do we do that? Well, we're going to do that by bending this lens. And so, this is the only structure that we can use to help bend our light, whether we're going to bend it closer to this fovea centralis. If we're reading something up close, we need to, so we can flatten it out or bend it. Here we're bending, here we're flattening it out. And so we can bend it to focus all the light source to our fovea centralis. And so accommodation outlines this idea, in which case the lens must change shape to focus on the objects. And to the fovea centralis. Okay, and direct that light to that fovea centralis. So bringing all that light source of that image towards your fovea centralis. Now, what happens once we bring that image to our fovea centralis? There's a couple of things, and we actually form what's called a real image. And this real image is formed on the retina as a result of bending that light. And so right now what you see, this word pathway, light, through, we see everything as upright. Okay, and whatever font size it is, we kind of know it's the standard 22, 23 size, Times New Roman's font or whatever. But in actuality, how this gets processed in the back of the eye is going to be very different. We form these real images. They're going to look reversed from left to right. So flipped horizontally. In addition, they're going to appear upside down. You can see that here, and of course flipped horizontally. And then and the real images are going to be smaller than the object. Which of course would be clear, right? So if you here, we see this as a font 40. Within our eyes, with our fovea centralis, it could probably appear as font size 10. Now that we've taken all the sensory input to the back of our eye at that fovea centralis, how does this get processed within our brain? And so we're going to take our optic nerve. There's actually 2 regions. We have the lateral region of the optic nerve, and we have the medial region of the optic nerve. Now what we're going to notice is that both medial regions from each eye of the optic nerves are going to cross over at this point, and this is called the optic chiasma. And before you write anything down, I just want you to just take a look really quick at what's going to go on, what's going to happen. So the lateral optic nerve from one eye is going to come with the medial optic nerve of the other eye, and they're going to come together at one point, being the thalamus. From the thalamus, actually taking a step back, what is the function of the thalamus? Good. And so it's going to start directing all that sensory input to the different regions of the brain. And so our thalamus, because we're dealing with vision and sight, we're going to direct all that sensory input to our occipital lobe. Taking a look at the right side, same thing. We have this lateral region of the nerve. It's going to come together with the medial region of the left eye, come together forming this optic tract. Collectively they form the optic tract. They're going to stop right here at this thalamus, where they're going to get redirected towards the occipital lobe for interpretation. So let's write all that down. So this optic chiasma, this is a location where the optic nerves cross. And we're going to have fibers from that medial side of each eye are going to cross over. Second thing we looked at already is the optic tract, and this is where the synapse with neurons occurs in the thalamus. And then collectively, the say synapse here, both of this are going to form the optic radiation, and this optic radiation is going to run from that thalamus towards your occipital lobe of the brain. And what is this purpose again of the occipital lobe? This is for visual interpretation and seeing. Now let's take a look at exactly the 3 different types of conditions your eyes can have. Whether you can see normally, whether or not the shape of your eye is distorted, in which case, once you try focusing your lens on that fovea centralis, it doesn't focus it under normal conditions. So metropia, this is the very first one we're going to look at, and this is when the eye focuses images correctly on the retina. And here's an emmetropic eye. Here you're going to have all that light coming in through your cornea. Your lens can easily bend all that light towards your fovea centralis. Now myopia, we also know this as nearsightedness. In which case, a person who has nearsightedness can't see far. And I remember this because it's my condition. I'm actually nearsighted. And when somebody has myopia, what happens to the light? Well, the light from these objects fail to reach the retina. Oh, so I'm sorry, this is one over here. So it's going to fail to reach the retina. And why is that? And it's going to actually focus all this light source in front of the retina. And this results if your eyeball is too big. So I got big old eyeballs, right? So eyeball shape are too large. So the light, what happens? The light from those objects fail to reach the retina. And are then focused in front of it. And because of this nearsightedness, objects in the distance appear blurry. Now how do you correct this? Well, using a concave lens that'll help bend the light and push it much further, so help focus all that image within that fovea centralis. Hyperopia, this is farsightedness. And this is when you can't see objects near. And so the distant objects are focused behind the retina. And as you can see here, this results if the eyeball is too small. So it's going to try to focus it normally. and it focuses it back here. And to correct this using a convex lens, we can bend the light, assist our lens in bending that light, and focus that image more anteriorly. Now astigmatism, actually I wrote one more thing. So hyperopia, too small, can't see near. And that being the case, near objects are blurry. And distant objects are clear. Now, if the eyeball is too short, or if you have a lazy lens, in which case, if we're trying to contract and flatten out or round out your lens by the ciliary body, that can give rise to that lazy lens. So it's not responding to that. Now, astigmatism, this results from light focusing as lines and not as points. And so looking at back here, instead of focusing the lines like that, all astigmatism, the lines are focused like that, so parallel lines. And the retina, and it's because of an unequal curvatures on the cornea or lens. And when the images appear, they appear blurry. And so I had a few students who said they have astigmatism, and their glasses are actually designed to have both concave and convex regions. So pretty interesting. Now, the last time you went to a doctor's office or optometry office, the one thing you probably were asked to do was to read a set of letters down a line, and they told you probably to keep on going down, down, down until you can't read a line anymore. And so this deals with one's visual acuity, and that deals with the sharpness or clearness of someone's vision. Okay, so what does this value mean? So 20 over 20, 20 over 20. The numerator is this value over here, and this is the feet at which the screening is done. And so that deals with U. Now the denominator is the line that you're going to determine, and that's the smallest value or the largest value that you get is going to be the line at which majority of the symbols are correctly identified. So what does that mean? Let's say that you have 20 over 60, meaning that your screening from the area that you were to where the board was let's just say 20 feet. Okay, so this is that distance, that's the numerator. Now 60 means that at 60 feet, someone who has great vision at 60 feet can see exactly what you've seen at 20. So meaning that the bigger the number this is, that means that there may be a more impairment within your vision. So someone who has 20 over, let's say, 200, that means that what you see at 20 feet is what someone at 200 feet can see. Or of course, not really 200 feet, but you get what I'm saying. That can mean that you don't really have that great of a vision. Okay, so those are what this numerator and denominator mean. Let's continue on to the next sense, and we're going to look at smell. And smell, we deal with what's called olfactory, so olfactory receptors. And these are neurons with long cilia. And do they have mucus? Yes. Now, chemicals, they have chemicals because in order for us to smell, chemicals need to be dissolved in solution. for us to Now looking at these impulses, so here's the roof of our nasal cavity. This is where we're going to find our olfactory receptors. And all these olfactory receptors, we always notice we have lots of this mucus here too. So we have this nice mucous membrane. Anytime you smell and take in any senses, you give off these smaller molecules and particles in the chemical form. That's going to get dissolved into this mucus, and that in itself is going to bind to receptors here within those dendrites of all of these neurons. So these are all these olfactory receptor cells. Okay, so they're going to bind to these receptors, and depending on whatever that chemical is, it's going to initiate a chain of reaction, send that action potential down, and then send that to the cortex. So which is this going to go through? It's going to go through the olfactory nerve. Okay. You get that in smell by our cortex. Now what we're also going to see is that smell is not in isolation. So ability to discriminate between smells goes hand in hand with another special sense, and that's going to be taste. When we talk about taste, we're really talking about taste buds, and when we look at the surface of our tongue, we have lots of these projections that we assume are taste buds, but in actuality, all these projections are called papillae. Some of them do have taste bud receptors and some of them don't. And so majority of our tongue that we see on the surface that don't have these larger bumps on them, these are called filiform papillae, and they're sharp with no taste buds. Then we have fingiform papillae, which are these ones, these much larger projections over here. These are rounded with taste buds. And then finally we have circumvallate papillae. These are the large papillae with taste buds as well. And those are these larger guys back here. Now what you want to notice also is, here's an example of looking at the circumvallate papillae right over here, is that all of these taste bud receptors are housed laterally. So they're found on the sides of each of the papillae. And so this is going to be the same thing for our fingiform papillae. And so what I want you to know, especially for your exam, is which ones have taste buds and which ones don't. Looking at our specialized receptor cells, so we have gustatory hair cells. These are spanning the very surface of our tongue. And then what happens? Well, we need mucus in order for us to dissolve all those chemicals. And in turn, that's all going to get picked up by these gustatory hair cells. Okay, so gustatory cells, these are our taste receptors. And these hairs are stimulated by chemicals dissolved in saliva. And looking at this location of our taste buds, we did cover the ones that are found within our tongue, but there are also other taste buds found within the soft palates, and then also our cheeks. And right now I just want you to pause and feel the roof of your mouth. Anteriorly, you put your tongue against that hard area, that's the hard palate, but put it closer towards your throat. Just run your tongue closer towards more posteriorly, and you'll notice that soft area. So that's the soft palate. Okay, gustatory complex. This is the region of the cortex that's going to receive those impulses by several cranial nerves. So the first one we have is the facial nerve. Facial nerve, this is going to be taking all that sensory taste input from our anterior tongue. And once again, that's cranial nerve number 7. Anterior tongue. Second we have is glossopharyngeal nerve. So that's cranial nerve number 9. Those good vibes from Vin Diesel. 3, then we have the vagus nerve. And this is cranial nerve number 10. And this is going to take in sensory input from our epiglottis. Number 2 is going to take information from our posterior tongue. looking at the different taste sensations. And so thinking about our tastes, we have 5 different receptors for 5 major taste categories. We have the sweet receptors, and these are for sugar, so saccharins, some amino acids. Those are found more anterior to our tongue. Sour receptors, these are what we consider as acidic. So anything that's acidic, like your lemons, very citrus, a lot of the citrus fruits that are very acidic. So sour receptors, these are found more laterally. Bitter receptors, these are more alkaline, so alkaloids, found more posterior. And then umami, this is a kind of like a meaty flavor. It also considers like that MSG flavor taste. So meaty, and that's found within your entire surface of your tongue. So protein, a lot of that protein too. Then you have salty receptors, and these are for metal ions. You think of like sodium chloride. And so salty is also found more anteriorly. And so a good example in try to determining where these receptors are is if you take like Sour Patches or candy that have both sweet and sour on it and salty, you can put it on your tongue and to see where those flavors are getting picked up all over your tongue. Now, earlier I mentioned that taste and smell do not work in isolation. They actually work together. And if you ever notice, whenever you get sick, you have a stuffy nose, and what happens to your ability to taste? It actually decreases. And so you can't have that discriminating taste as well. And so why is this? Well, let's take a look at, compare and contrast these two different senses. Both of these senses use chemoreceptors, in which case they're going to have that chemical dissolved, whether it's going to be in saliva or within that mucus within the nose. And it's stimulated by these chemicals, both found within solution. Taste, we have 4 different types of receptors. Well, I'm sorry, we actually have 5. I mentioned umami. And then smell, we have a large range of chemicals that can differentiate. And because it can differentiate a lot, it's not just limited to these 5 receptors. Now, both of these senses will complement each other and respond to many of the same stimuli. Now, if you imagine, so if every time you chew, what happens? As you chew, you're actually expelling a lot of that flavor found within your mouth into your nasal cavity. And so here's my bad cross section. That is the nose. Here is the mouth, the neck, back of the head. Here's the oral cavity. Here we have the pharynx. And as we remember, it's really bad, right? I know. Here's our nasal cavity. Here's our oral cavity. And we can join them together by this pharynx. Here's the pharynx. So as you eat, okay, I'll just draw the eyes so you kind of have a sense of what I'm looking at. Oh, and the hair. So draw the hair. Okay, so here as you exhale and as you chew, you're bringing some of these chemicals into your nasal cavity. And because of that, those chemicals can also get picked up by all of those olfactory receptors in your superior region of your nasal cavity, giving rise to this complex flavor that you get as you eat. And so you notice sometimes people who also, when they drink that wine, what do they do first? They swirl it, they smell it, and then they eat it. And then they drink it, not eat it, but then they drink it. Okay, so it's this interplay that allows you to have really grasp complex flavors. Now we're going to stop here, and in the next video lecture, we're going to go over hearing and equilibrium.